Hi, I'm Julie Wolf with IndieBio, and we're here with Founder Stories, where we learn a little bit about the motivations and the technologies behind the IndieBio founders and their companies. I'm joined today by Adam Kirkowitz, CEO of Biomage. Thanks for coming and joining me today, Adam. I appreciate you taking the time.
Hi, Julie. Thank you so much for having me.
Biomage is a company that will turn every biologist into a bioinformatician. But I think in order to understand what Biomage offers, we first need to understand the technology and the techniques that it supports, namely single-cell RNA sequencing, sometimes called single-cell transcriptomics. What can you tell me about the scientists that use this technology and how it's used in order to make different discoveries?
Sure. So single-cell transcriptomics or single-cell RNA-seq is a relatively recently discovered method to really understand what's happening inside living organisms at the level of individual cells. And this is something that I like to compare to the invention of light microscope a few hundred years ago, when scientists for the first time were able to look at individual cells from the outside. And single cell gives us that perspective and that ability to to look at individual cells from the inside. So it's a unique capability that has only emerged in the past couple of years.
And so using this type of technique, you can use this technology to support a lot of different fields, correct? I imagine that there's people using this in developmental biology, in various disease states, et cetera.
Yeah, correct. So the technology is not specific to just one type of biological research or biological endeavor. It has been used throughout many different fields of biology, including prominently cancer research, cardiovascular research, and also research into discovering new cell types. So developmental biology, discovering how organisms develop, and new things about organisms that we haven't known before. So a good example here would be a discovery of new type of lung cell that was done at Harvard Medical School, and this new type of lung cell, the pulmonary ionocyte, was immediately linked with the rare disorder cystic fibrosis, pointing at potential ways to cure that disease.
Oh, that's very interesting. And I guess that this pulmonary ionocyte must not have looked different. So using that example of the microscope, you couldn't look at it and say, this is a different type of cell, but by investigating the RNA profile of that cell, people could differentiate it from its neighbors. Is that right?
I'm not familiar with the exact details of this particular research, but certainly looking by analogy at other types of science that was done using single cell, we've seen discovery of new cell types where we already thought that we knew all the cell types. So further subdivision of cell types that have previously been thought to be homogeneous. That certainly did happen in the past couple of years using this technology.
I see. And since this is a relatively new technology, if one looks at PubMed or does a literature search for something like RNA single cell sequencing, you can see that the number of publications is growing. I don't know if you could actually say exponentially, but it's growing quite a bit, right? So how do you see this technology or this technique being used going forward as well?
Yes, so I think going forward, perhaps one of the biggest challenges, specifically due to this, as you described it, exponential interest in the technology, is perhaps linked with the fact that it's going to be really difficult to scale this technology to allow every single biologist to use it. So this somewhat links to the work we're doing here at Biomage, where we're actually making it possible for every biologist to analyze a single cell data set without having to develop the type of really elitist expertise that has been required so far to be able to carry out such analysis. So in a sense, what we do at Biomage is we precisely allow biologists to cope with this exponential growth and the and the demand for this technology, and we help to bring it to every biologist who wants to use it.
Absolutely. So in this case, the same biologist who's working at the wet bench, preparing the tissues, extracting the RNA, and I imagine still sending it off for sequencing, although I guess in some cases they could perform the sequencing themselves. When the sequencing is done, that same biologist can directly look at the files, correctly?
Yeah, so the idea is that we are removing effectively a step in the process where the files that were created, the files that were created from analysis of a sample of a tissue have to be first worked on by a research bioinformatician. So we remove that step entirely. We're effectively automating research bioinformatician, and we're making it possible for biologists to become bioinformaticians themselves, as you very neatly put it in the beginning of this interview.
And I imagine that this will free up all biologists to start working on their projects and not have that bioinformatician be the gatekeeper, so to speak, of the analyses for all of the individual experiments. Is that the plan for Biomage?
So certainly, so the benefits are not just cost efficiency, it's also a quicker learning loop and a quicker iteration. Biologists can test their hypothesis directly by directly looking at the data set without having first to communicate it to another professional who will deliver an answer to them once they've got the time to actually action it. We remove a lot of potential issues with miscommunication and knowledge transfer between these two very different fields, that of biology and that of bioinformatics.
And so how does the conventional route of analysis versus Biomage and its software platform compare in terms of time that it would take to get a result?
In a typical single cell analysis, a bioinformatician working part-time would take between 3 to 6 months to deliver the level of insight that is required for a publication in a high-profile journal. And our aim, our goal as a company is to bring that process to just about a week or two of hands-on analysis by the biologist directly with the software. So
Wow, that's a lot faster.
we're significantly cutting down the amount of time that's required to carry out such analysis.
And you mentioned there's a cost benefit. Does that mostly come from not requiring the labor of the bioinformatician expert?
Yeah, that's correct. So so the bulk of the cost saving is specifically not having to rely on a consulting service or potentially a partnership with a qualified bioinformatician. There is some additional cost and cost reduction in how we handle the data and how we can process the data directly by a close integration with the place where sequencing actually happens with a core facility. So by directly integrating with core facilities, we can make it more cost effective to process the data and to actually carry out the computational aspects of the analysis as well.
Oh, very interesting. Now you've been working with a number of core facilities showing your initial product and working out some of the bugs. I wonder what the response has been from some of the bioinformaticians working in those facilities. For example, are they a little worried that you're going to put them out of a job?
Yeah, I get this question quite a lot, and actually, interestingly, not from research bioinformaticians themselves. They're very happy to partner with us because working together, we can actually deliver the biggest value to their customers, and we can make them available to work on the truly creative and truly difficult aspects of the field. So just to give you a comparison on in an average sized core facility. There will be typically two research bioinformaticians who typically be taking care of as many as 50 projects. They really do need an ability to to cope with the analytical needs of that many projects, and effectively by bringing down the time to analysis to 1 or 2 weeks, we make it possible for these bioinformaticians to effectively do their job. So they're actually very happy to partner with us, and the initial reception from core facilities on both sides of the Atlantic, both in the UK and in the US, has been overwhelmingly positive, and we're expecting these partnerships to further expand into other core facilities, and to grow stronger by more and more closely integrating with these core facilities, with these partners.
Oh, it's very good to hear that they are happy to partner with you. And these core facilities, they're found at most of the major research institutions in the UK, in Europe, in the US. Are there other places where you could imagine your software being useful as your company continues to grow? Other spots, perhaps in the private sector?
Yes. So these core facilities, we consider them to be the academic market, whereas we think that the bulk of the users and the real impact of the software that we're building is going to most likely come from other sectors, including pharmaceutical research and including biotechs. So our plan is to initially treat the academic customers as a way to validate our technology and as a way to get an initial beachhead into this space. And it will be for the next stage that we're going to target biotechs and pharmaceutical companies. They're the next type of customer for us, where we think the true impact of our technology is going to be.
Well, that will be very exciting to see. Now, you clearly understand the biology. You have a bit of biology in your background, but you're a very strong bioinformatician and have a strong programming background. So I wonder how you recognize that this was a problem that even needed to be solved.
Yes. So my journey started on the side of programming, computer programming and mathematics. That was the focus of my undergraduate degree. Following that, I worked for a short time in Skyscanner, who are a software company where I understood what software engineering is like, and understood how software can be used to solve real life issues and help people to accomplish things, accomplish the tasks, get their jobs done. The biological side that came a bit later. I realized that in order to have a really impactful professional life, I really wanted to come a bit closer to human health and a bit closer to understanding how biology works. So I enrolled in a PhD program at the University of Glasgow, where I ended up doing quite a lot of bioinformatics, actually. I managed to find a way to merge my my scientific interests with my software engineering interests, and that is the sort of expertise I developed at the University of Glasgow. Going back to your question, how did I realize that there was such a need? Actually, while a PhD student, I realized that there was a really big need for bioinformatics analysis among biologists that I worked with. So I started offering such analysis as a consulting service with the two co-founders in Biomage, Marcel and Eva. And very quickly we realized that it will be impossible for us to cope with the demand. And the most impactful way to allocate our efforts was actually towards building a software solution to solve that difficult problem of allowing biologists to understand single cell data.
I see. So you were in an academic institution. You've worked with top academics, both bioinformaticians and biologists. What motivated you to take this down the entrepreneurial route rather than to continue to develop a software in a lab or some other academic setting?
So a really big challenge for developing software in an academic setting is the fact that it's so very difficult to scale a solution and to bring a solution to market within the fairly rigid framework of academic grants. So grant money is typically not dedicated for software development. It's typically dedicated for discovering new science. And the fact that in order to discover that new science, scientists need excellent software, that's very often treated as an afterthought or something that can be only a small part of a research grant. And this is why through conversations with senior colleagues in academia, I realized that the best way to realize that mission, the mission of building really great software that can help make biologists more productive, the best way to realize that mission was as a private venture.
I see. Well, that sounds great. And it sounds like you'll be solving a problem that many people have by creating this software platform. Now, you already mentioned you have two co-founders, Marcel and Eva. Can you tell me a little bit about how the three of you came together and decided to found Biomage?
Sure. So actually our story, the story of how we met, is interesting in this aspect that we didn't meet actually in the same time, but we did meet in the same place. So this place was the undergraduate laboratory at the University of Glasgow, the computing lab at the University of Glasgow, and in the building called Boydor Building, which is actually a subject of many jokes among the students, because University of Glasgow is actually a very beautiful university, and the main campus building looks like a castle, and I think at some stage was even supposed to become part of the set for Harry Potter.
Wow.
And so it appears in all postcards and all promotional material of the university. The actual place of study, which is the Boydor Building, is nowhere near as pretty.
Not on any postcards.
It's a brutalist concrete building. So in that very brutalist concrete building, I've met Eva first when I was an undergraduate student at the University of Glasgow, and she was a student in a different class. So we started talking, and eventually a strong relationship emerged. Marcel I met a couple of years later when I was in a different position, not as a student but as a teacher. So I was tutoring an undergraduate lab, and Marcel was one of the undergraduates. He was exceptionally gifted, and really I often like to say that it wasn't me teaching him, it was him teaching me. So so I knew we had to work together. So that was the original co-founding team of Biomage. And following the raise of our seed round, we'll be bringing in another founder with specific expertise and single cell sequencing. So it's going to be Professor Peter Karchenko from Harvard Medical School.
Oh, very nice. Yes, you had the good fortune to almost complete your seed round, and hopefully that by the time this video comes out, you'll even have closed your seed round during the program. In addition to hiring Peter, what will you be using those funds to do?
So the money that we've managed to raise, that we'll have managed to raise from our seed investment, we're going to be spending that on hiring a research bioinformatician and a full-time biologist who is going to act as our chief scientific officer with a substantial commercial role. So these two positions will be filled as soon as possible. We're looking to bring in another software developer as Eva, who's the chief technology officer in the startup, as she's taking more of a project managerial position.
Oh, Very exciting. Now I want to travel even further into the future, not just the immediate coming months, but maybe like 5 years or so. And I want you to imagine what you think the world looks like. How are scientists using Biomage's software platform? What other products might you have developed? And what does the company look like now that you're not such a startup phase?
Sure. So our goal, as you put it, a 5-year goal is maybe even sooner, maybe even within the next 3 years, we would be expecting um, to fully win or dominate the landscape for single cell data analytics. So in 3 years' time, we would like everybody to be able to use Biomage as their solution of choice for single cell data analytics, expanding into other technologies such as spatial transcriptomics, such as some of the dual kind of bimodal single cell RNA and DNA-seq. So the goal here really is just to make this technology as available as possible, while maintaining a strong commercial presence and commercial viability. So we really hope that we can make this solution very accessible to academic community, and we can cover the commercial needs of the company by working by partnering with pharmaceutical companies. So we hope that the bulk of the revenue is not going to be coming from the academic market, but from biotechs and from pharmaceutical companies.
Well, it's a wonderfully ambitious plan, and I'm happy to support you in it. And I'd like to thank you. I know, like all of our IndieBio founders, you're extraordinarily busy. I'd like to thank you for taking time to speak with us about your journey and your founder story.
Thank you so much, Julie. It's been a pleasure to talk to you.
For more founder stories, please visit indiebio.co/founder_stories. And to apply to be a part of IndieBio's next cohort, please visit indiebio.co/apply.